effects, and birds. Okay, so for our first speaker, to kick things off, is going to be Jason Godfrey, who's going to talk about salmon of Scottish coastal waters, recent advancements in knowledge in relation to their interactions with marine renewable energy installations.
A snappy title, I'm sure you'll agree. This is some work that we did last year with colleagues in Marine Scotland Science. Not sure what happened there. So the background you're probably all very familiar with, that the Scottish Government has some very ambitious targets for the development of marine renewable energy. The question is, can it be done sustainably? There are certain legal obligations, the EU Habitats Directive, to be met, for example. And what's needed is information about possible interaction between animals, in particular migratory animals, and developments. So the animal that we're concerned with here is the Atlantic salmon, which is a species of high cultural and conservation value, and has several SACs designated. So the starting point of kind of all these studies is information about the spatial overlap between the species of interest and the energy developments. So briefly about the salmon life cycle. Salmon spawn in rivers and where they grow for a couple of years before migrating to sea, where they will grow for 2 further years before returning as adults. And it's this section in the sea that concerns marine renewable developments, and in particular I'll be focusing exclusively on the returning adults, about which very little is known. What's presently known about inshore migration of returning adults is that they return throughout the year, but there's a peak of numbers in the summer, but very valuable stocks return in spring. And all the information that we've had until now has been from physical tagging with known capture and recapture points. So for example, fish tagged here at the South Esk may appear anywhere on that coast, indicated by the blue dots there. But the overall picture is very complex. Fish may be swimming in either directions. All we know really is that they return to the coast and travel to the river of origin, but there are no consistent patterns. But there are significant limitations to this approach because they only have the start and end points. That kind of constrains us to thinking about the species as being coastal in its movements, which may not be true. So an alternative to using this sort of capture-recapture is to use satellite tags. We're familiar with fantastic studies of birds, usually big birds, that can carry satellite tags where they provide fantastic tracks of where they've been throughout the world. But there's a drawback in using satellite tags with fish, which is that the tags don't transmit underwater. But they can be programmed to record what happens, depth data, temperature data and so forth, and programmed to pop up after a certain period. They must have buoyancy, at which point they can start communicating with the satellite, upload the data they've recorded, and give a single positional location. They have about 2 weeks to tell us everything that they have stored in their tags before the battery runs out. So the study site we used is Armadale, which, as you probably know, is on the north coast of Scotland, around about there. It's just within the Pentland Firth and Orkney waters area, and very close to the proposed development at Far Point of wave power. And the boat you can see in the picture there is a commercial netsman's fishing boat. It's also our laboratory. The netsmen use a traditional method called bag nets, which also used in Norway. They're kind of large triangular-shaped nets, about 3 meters high, 3 meters wide. And they have a leader which draws fish into a sort of lobster pod arrangement of successive baffles, where the fish is stored in the end piece and can swim freely within them, so they're captured in really good conditions. And here's a picture of the net, the head pole of the net, and the leader line leading into the trawler. So we did all the tagging on board the boat. It's about a 3-minute procedure, 2-hour recovery period, and we used 1 to 10 day deployments for our fish. We also took a tissue sample from the fish for subsequent identification of region of origin. So here's a wee video, which I hope will work. Oh dear. Any ideas? Click on it, let's say. This is a video of the salmon being released, a salmon being released. I think it is now. Oh dear, doesn't seem to be working very well. Well, you just have to imagine a beautiful video of a fish swimming off with a tag on its back, and that's the last we see of the fish, and everything else we know comes from the satellite data. Now, you saw another version of this map earlier on in Ian's talk, and he described this as quite a broad scatter, which I guess is fairly true. But the question is, are these salmon in the area relevant to marine renewable developments? And I think we can say that these are all very close to the vicinity of marine renewable energy sites that we've seen mentioned today. We got about 44 tags out of 50, which was much better than we'd expected. And the tags are probably mostly in coastal zones, but they're appearing up to 100 kilometres offshore, with minimum daily travel rates of up to 55 kilometres an hour. But what this map doesn't show us is where these fish are going. And that's kind of crucial towards picking out pathways and understanding what populations from special areas of conservation, for example, where they may be swimming. So we need some help to achieve that. We use genetic assignment techniques, of which a number are available. They all exploit the strong spatial variation in salmon DNA, and it's strong because they're homing creatures. The particular technique we use is the single nucleotide polymorphisms, or SNPs, not SNP. And these are used where a single base pair varies in a long, otherwise identical sequence. And through this, we can characterize regions or rivers based on the frequency of these SNP variations within the population. But first, you have to set up a baseline to describe the population. So we've got 4,000 fish and 150 sites spread right across Scotland, Northern England. And we examined sort of 5,000 sets of SNPs for each of these. So it's a huge data set. We analyze it using hierarchical cluster analysis to identify assignment regions with similar genetic signatures. And then we identify which are the best SNPs to use for subsequent work. So this is very much work in progress, and at the moment we're only confident in identifying sort of 4 populations within Scotland, broadly shown by that map there. We can get more detailed than that now, but we're not ready to present that. And if we look at that map of the fish that popped up earlier, we can see, well, yes, maybe the fish that are bound for the east are sort of heading north and east around Orkney, and there's some sort of collection of the northwest fish in the northwest, but there are lots of exceptions to the expected pattern, so we're not seeing fish that are necessarily heading straight to their river of origin at this moment. We also have a number of unidentified fish there, some of which are from Norway, from Ireland, or possibly from other parts of Scotland. But it's a work in progress. We need to be collecting more data before we can be identifying to river level assignments, which is what we're really interested in. So we've established that these are fish that we're interested in, and we've established an area that we're interested in. So now we need to know about what sort of depths they're swimming at to determine whether they're likely to interact with devices or not. And we need time and depth profiles to parameterize models of strike risk. So here's an example track of salmon, which I'll talk through a little so you understand it. The grey bars represent periods of darkness, so this is about an 8-day period. The red is the temperature, and the blue is the depth to which the fish is diving. You see a period of diving initially, then a period of the fish at the surface, then progressively deeper dives, before the fish again returns to a period at the surface, and at the same time the temperature increases there, and that's actually a fish entering a river, actually the River Neva. And that black arrow there points to the point where we cut off the data. We don't use the data to the right, we're only interested in the data to the left. Here's another example. This is much more patchy data. You can see a lot of gaps in the data. And we're slightly puzzled by the break that occurs halfway through the sequence there, and we thought maybe it's a tag malfunction. But we were lucky enough to recover this tag. It was returned to us by a member of the public, and it contains on it two bits of data. One, a huge data set that's stored within the tag itself, and two, on the outside of it, it bore the marks of strong animals' teeth. And we ended up working out that this was a fish that had been predated almost certainly by an endothermic shark, because it's a stomach temperature of around 25. So again, we dismiss the data to the right and we use the data to the left. But that's not the only thing. That's not the only thing that salmon have to look out for, because this is another example of predation event. This time it's a mammal. It's 37-degree temperature. So it's not just marine renewables that some have to look out for. So when, not surprisingly, we see a lot of variation between individuals, as we would expect, but if we analyse the data in some detail, again using cluster analysis, we can detect three sort of groups of behaviours. One indicated in the top panel of that graph is very shallow surface-orientated fish, but nevertheless undertaking brief dives to depths of maybe 100 metres. Then at the bottom you've got a fish that's actually spending several hours at 60 or 80 metres of depth and returning to the surface only for brief time periods. In the middle, something in between. So another way of looking at this is to look at a depth class profile, and there's three individual fish there, and you can see one that's spending, a surface-orientated fish that's spending maybe up to 80% of its time in the top 5 metres. At the other end of the scale, you've got a fish that's only spending 25% of its time in the top 5 metres and substantial periods of time down at 50 metres. So what can we conclude from all this? Well, firstly, that salmon are not necessarily migrating around the coast, but they can be many varying distances offshore. It's not clear in which directions that they're heading. But they do predominantly inhabit the upper surface layers. Individuals vary, maybe populations vary. We need to explore that further, and we'll be doing so, I think. But all individuals undertake dives, and the depths that all the individuals reach imply that they're using the whole of the water column. Further, individuals from various populations may be present in zones with marine renewable energy development, and so none of the fish swimming there can be dismissed as less important animals. And finally, salmon are most likely to interact with surface devices, but could come into contact with any device. But of course, whether they're damaged by these devices, that's a whole different question that we haven't provided any insight into. So for next year, or rather this year now, this summer, we'll be tagging further salmon and using longer tag deployments, hopefully tease out some of that variation amongst the data set, and also we hope to complete the genetics work, maybe getting down to a river level so we can identify the populations in which they come. Thank you.
Jason's going perfectly to time, so if there's any questions out there, we've got a couple of minutes. Can I ask Jason, on you? Yeah. If a fish, you've got these dials on the tags, if you can call them that, the descent to deeper water. Got any idea whether they're going benthic? Can you tell from the profile or the information?
Absolutely. Yes, the answer is at least on fish are this one fish died, and you can tell it was dead because it landed on the surface, and there was a tidal pattern showing it was stuck on the surface. And shortly before its death, it was spending time at that same depth. So within that, it was swimming to the bottom, spending a bit of time there. Some will just go down and touch, come up, some spend time. Who knows if they're feeding or avoiding predators.
Yes. Good to go.
All the satellite records were in Scottish waters, and yet I caught so many salmon went as far as Iceland and Greenland. Is that just bad luck that you didn't get any of those?
No. These are fish that have already been in Iceland, probably. Come back and we caught them on the coast, and they just have short deployments around the coast. So